This is a revision video for AQA GCSE Combined Science Biology Paper 2. It covers the topics of homeostasis, genetics and evolution, and ecology. The first topic in Biology Paper 2 is homeostasis and response. Homeostasis is the regulation of the internal conditions of either a cell or a whole organism to maintain the optimum conditions for function in response to internal and external changes. What that means is that the environment around you is changing all the time. So your body responds to that by making sure that inside a cell, the conditions stay the way you want them to. So the temperature stays the same, the water content stays the same, all that sort of thing. Often, this is controlled by negative feedback loops. So a negative feedback loop is just where your body will make enough of a hormone or enough of an enzyme until eventually you say, okay, that's enough, that's too much, and then it's inhibited and it stops being made. The three examples we need to be able to talk about for the GCSE biology are blood glucose concentration, body temperature, and water levels. It's really important that the level of water in the blood is controlled, because if it isn't, then the red blood cells could either shrivel up or burst. We naturally take in water when we eat and drink, but then we lose some in an uncontrolled way when we breathe out and also when we sweat, but also in a really closely controlled way from our kidneys. In the kidneys, that water is used to dilute urea, which is toxic, and it's come from the breakdown of proteins and their deamination from toxic ammonia. The way that homeostasis works is that a type of cell called a receptor detects a stimulus. So a stimulus could be the temperature changes, or it could be suddenly there's more light. Then a coordination centre like the brain, the spinal cord, or the pancreas says, okay, we're going to take that stimulus and we need to respond to it. So it's then going to pass some kind of signal to an effector, which could be a muscle or a gland, and that's going to have a response. So there are nervous responses from your nervous system, and there are also chemical responses from your endocrine system, which is the hormones, and also there are some responses that involve both of those systems. So you might be wondering, why do we have these two systems? Well, the endocrine system works using hormones which are released from glands, and it's slower and produces longer lasting responses. So your endocrine system is responsible for things like you going through puberty, but also for responses like dehydration, where it needs to go on for quite a long time. Your nervous system obviously uses nerves or neurons, and they transfer electrical impulses. Now those responses are very, very fast, but they're also very, very short-lived. So they're useful for things like if you touch something hot and you burn yourself, you need to do a reflex reaction and move your hand straight away, but you don't need to keep on responding after that. The nervous system is made up of receptors, such as the retina cells in your eye or your skin, and it passes information in the form of electrical impulses to the central nervous system, or CNS. This is a combination of your brain and your spinal cord. The CNS then coordinates the response of the effectors, so that could be muscles or it could also be glands. Between those nerves or neurons, there are little gaps called synapses. Now, when an electrical signal has travelled down a nerve and it needs to get to the next nerve, it has to cross that synapse. And it does that by releasing a chemical called a neurotransmitter, which can diffuse across the gap between the two neurons. And this will then trigger the next electrical impulse. So when you think about how drugs work and the fact that, say, if you drink some caffeine, then you seem to be able to move faster and think faster, or if you have some alcohol, you're slowed down, often those drugs are affecting the speed at which neurotransmitters are being released or are travelling. A reflex reaction is a rapid, automatic reaction. It doesn't involve the conscious part of your brain, so you've never had to learn how to do it and you don't think about it. You were just born with reflexes. So reflexes tend to be all about stopping harm happening to your body. So it could be something like a reflex response to you touching something hot or to a very bright light. You need to be able to discuss how a reflex reaction works. So firstly, there's a stimulus. That's the thing that makes the reflex reaction happen. What has changed in the environment? So say you could have the stimulus, which is a change in temperature, is detected by a receptor. So for a temperature change, it would be in your skin cells. For light, it would be in the retina cells of your eye. The receptor then generates a nervous impulse, so that's an electrical signal, that passes along the sensory neuron to the relay neuron. So you've got three types of neuron in this reflex response, and you need to know what they are. So the sensory neuron is connected to the receptor. It's the thing that's doing the sensing. The relay neuron is going to pass the electrical impulse through your spinal cord. Then it's going to relay it on to the motor neuron. So motor is all about movement. This is the neuron that's going to be responsible for passing it on to the effector, which is going to cause your body to respond. So here, the effector might be a muscle in your arm which causes you to move your hand out of the way so that you don't get burned. The first required practical in Biology Paper 2 is all about reflex reactions. You're going to investigate the effect of a factor like caffeine or tiredness on how quickly you can complete a task like dropping a ruler. In the ruler drop test, you're going to use a conversion table to turn the distance that the ruler has travelled before you manage to catch it into a reaction time. And you're going to do this experiment in 2 different occasions, one just as a control, and then a second one where you've changed a factor, like drinking several cups of strong coffee. You're going to use a conversion table that allows you to translate the distance that the ruler has fallen before you catch it into a reaction time. And you're going to need to control certain factors, like always using the same hand, because the hand that you write with, you're going to have better dexterity, so maybe you'll be able to catch the ruler faster. Sometimes you could do an alternative method where you use some kind of computer program and just clicking. Now that's more precise and it's going to be timed to a greater resolution, but it might not be available for everybody. So in doing this in a class situation, it's probably not a good option. The endocrine system is the second system that's involved in homeostasis. Endocrine glands are cells that can secrete hormones. These are chemical messengers that are carried in your blood until they get to a target organ where the hormone needs to act. There are 6 specific endocrine glands that you need to know the locations of. The pituitary gland, that master gland, is located in your brain. Your pancreas, which is part of your digestive system, is sort of in the middle of your abdomen. Your thyroid gland is in your neck. Your 2 adrenal glands are sat just on top of your kidneys. And then if you're female, you have ovaries, which are a little bit lower than that. And if you're male, you have testes, and you know where those are. The pituitary gland is called the master gland because it's a gland that controls other glands. It secretes hormones into the blood, and those hormones will then act on these other glands to stimulate other hormones to be released. Your thyroid produces a hormone called thyroxine, and that stimulates your basal metabolic rate, so basically your underlying metabolic rate the whole time, which is necessary for growth and for development. And it's controlled by a negative feedback loop. So you make some, and then eventually when the levels in your body rise high enough, that's detected, and then your thyroid gland will stop making it. Your adrenal glands produce adrenaline, which is something that you make in times of fear or stress, so it leads to that fight or flight response. It increases the heart rate, and the point of that is that it's going to increase the amount of oxygen and glucose that gets into your brain and to your muscles. It's important that your blood glucose levels remain constant, because without that, you're not going to have regular energy levels, and also it can cause damage to your cells. Your pancreas is responsible both for monitoring the blood glucose levels and for controlling them by releasing hormones. If your blood glucose gets too high, the pancreas makes a hormone called insulin. And what insulin does is it increases the permeability of your cells, particularly in the liver, and that causes glucose to move into those cells. Then that extra glucose can be converted into a polymer called glycogen for storage. If you're doing higher tier, you also need to know what happens when the blood glucose gets too low. So then the pancreas produces a second hormone called glucagon, and glucagon causes glycogen to be made back into glucose and released back into the blood. So here you can see the blood glucose level rises, and therefore the pancreas is stimulated. It produces and secretes insulin, which means releasing it into the bloodstream, and then that insulin stimulates glucose uptake from the blood, so the blood glucose level falls. Then, if you're doing higher, you need to know that this again will stimulate the pancreas, and therefore glucagon is secreted. And glucagon will stimulate the liver to release glucose, so the blood glucose level rises. Linked to blood glucose levels, we can think about type 1 and type 2 diabetes. So type 1 diabetes is a disorder where the pancreas isn't making enough insulin, and so it's characterised by uncontrolled high blood glucose levels, and you normally treat it with injections of insulin. You can diagnose it with the 4 T's: going to the toilet all the time, being thirsty all the time, being tired all the time, and losing weight. Type 2 diabetes is a little bit different. It's not that the body isn't making insulin, but it's that the cells are no longer responding to it. And generally this can be controlled by controlling the amount of carbohydrates that you're eating and maybe an exercise regime. And it's known that obesity is a risk factor for type 2 diabetes. Basically because if you constantly have high blood glucose levels because you're eating a lot, it's likely that eventually your cells will become desensitized to that insulin and they'll sort of start ignoring it. During puberty, the levels of reproductive hormones like estrogen and testosterone begin to rise, triggering secondary sexual characteristic development, such as breast growth and facial hair growth. In females, the main hormone is estrogen, which is produced in the ovaries, and this is linked to the beginning of ovulation, where eggs start to mature and be released. Testosterone is the main male reproductive hormone which is produced in the testes and stimulates sperm production. The menstrual cycle is controlled by 4 hormones. Follicle stimulating hormone, or FSH, causes an egg to mature and become ready for ovulation. Then luteinizing hormone, or LH, stimulates that egg to actually be released. The ovaries are responsible for making estrogen and progesterone, and they're both involved in maintaining the uterus lining. At the start of the menstrual cycle, as progesterone levels fall, the lining of the uterus is shed in menstruation. Rising levels of follicle-stimulating hormone cause the egg to mature, and rising levels of luteinizing hormone lead to ovulation. These 2 hormones also promote the production of estrogen and progesterone, and when they are produced, the release of FSH and LH is inhibited, which means it stops. High levels of progesterone mean the uterus lining is maintained in case the egg is fertilised, and then when progesterone levels fall, menstruation occurs again. When you're picking contraceptive methods, you need to think about how reliable it is and therefore how likely it is that you'll get pregnant, how easily available it is, whether there are any health implications, whether there are any ethical implications, and also how expensive it is. Hormonal contraceptive methods use the hormones that are produced by the ovaries to prevent the production of the hormones that are made by the pituitary gland. Those are responsible for causing eggs to mature and be released. Hormonal contraceptive methods include the pill, the coil, injections of progesterone, implants, and patches. If you're not using a hormonal method, which could be for all sorts of reasons, like the fact that some people are more susceptible to blood clots and estrogen can play a role in that, then there are non-hormonal methods that are available, such as condoms, what we call a barrier method, which is going to actually stop the sperm from getting to the egg, a diaphragm, and also spermicidal creams, which you can use with condoms and diaphragms, which are actually going to kill the sperm. Some couples struggle to get pregnant and they may undergo fertility treatment. The first stage of fertility treatment will involve taking a drug that contains FSH and LH. The idea of this is that this will stimulate egg maturation and ovulation if those just aren't happening, and then it may be possible for the woman to become pregnant in the usual way. If that's still not working, they may undergo in vitro fertilisation, which is basically where the sperm fertilises the egg in a petri dish. So the mother is given FSH and LH to stimulate the production of several eggs, and then they're collected and they're fertilised by sperm in the lab. Once those eggs start to develop into embryos, when the cells start dividing, several of them are inserted back into the mother's uterus. Although fertility treatment does mean you have a better chance of becoming pregnant, it's obviously very emotionally and also physically stressful, and the success rates aren't that high. And it can lead to multiple births because the process is so intensive that they tend to insert multiple embryos in one go in the hope that one of them will survive. But obviously sometimes several of them survive, so then it's more likely that you'll have twins, and that can be a risk both to the babies and the mother. The second unit in Paper 2 Biology is reproduction, and the first thing we need to know is all about the types of reproduction. So in sexual reproduction we have gametes, or sex cells, and the nuclei of those gametes are going to fuse to make genetically unique offspring. Usually it's going to involve 2 parents, although there are lots of plants and some animals that can self-fertilise, where one organism is producing both gametes itself. And there's still going to be variation because those gametes are going to have slightly different combinations of alleles in them. So sexual reproduction is going to produce offspring which are different to each other and different to the parents. Asexual reproduction is going to produce clones, or genetically identical individuals, and so there aren't any gametes involved. You need to know about 2 types of cell division. For paper 1 we talked about mitosis, which happens in all body cells and allows them to make more body cells for growth or for repair. It starts off with DNA doubling, and then the cells divide once, so there are 2 diploid cells made. These cells are identical to each other and identical to the cell they were made from. Meiosis we only talked about for paper 2, and it happens in the reproductive organs. So in humans, that's in the ovaries and the testicles. It makes gametes like eggs and sperm. The DNA doubles at the start, just like it does in mitosis, but then the cell divides twice, and that means that 4 cells are made, and those cells are haploid. They only have 1 copy of each chromosome, rather than the 2 copies that a diploid cell has. The cells are different from each other because there's different assortment of the chromosomes coming in, and they're also different from the original cell because they've only got half of its DNA. Those cells that are produced in meiosis are gametes, and they can then go on to join up with another gamete and produce a new cell which is back to the original amount of DNA. There's a lot of new vocabulary in Unit 6, and it's really important you're familiar with this, because quite often we see people who understand the science, but because they're not using the proper terminology, they don't get the credit for it. So, a gene is a length of DNA that codes for a particular combination of amino acids which join together to make a specific protein. That could be something like a pigment to make your hair brown. An allele then is the different variants of that same gene. So for instance, some people have the allele which allows them to make that pigment and therefore they have brown hair. Some people have a different allele for a faulty version of that pigment, so it's not actually brown and therefore they have blonde hair. So we talk about the brown hair allele and the blonde hair allele of the hair colour gene. Your genotype is the actual alleles that you have. So whether you have two brown hair alleles or a brown hair allele and a blonde hair allele. And then the phenotype is the overall effect. It's what is expressed, what we can actually see. Expressed is a really, really important word here, so you need to be familiar with that. We're not going to talk about alleles that show up. We want to say alleles that are expressed. So if I look at somebody, I can see that that person has brown hair. That is being expressed. That is their phenotype. But what I can't tell without doing a genetic test is whether they have two brown hair alleles or a brown hair allele and a blonde hair allele. So I can tell their phenotype just by looking at them, but to know their genotype, I need to do some genetic testing. When a person has two of the same allele, so two blonde hair alleles or two brown hair alleles, we call that being homozygous, and where they have different alleles, we call that being heterozygous. Now, when we look at all those alleles, a recessive allele is one that will only be expressed, it will only show up and be in the phenotype in the homozygote. So you need two copies of it for it to be expressed. A dominant allele is one that is expressed even when there's only one copy there, so even in the heterozygote. And I can't emphasise enough how important it is that when describing recessive and dominant, you use the word expressed. The genetic material of a eukaryotic cell, like a human cell, is contained in the nucleus, and it's made of a chemical called DNA, or deoxyribonucleic acid. DNA is an incredibly long molecule. It's made of two long polymer chains, which together form what's called a double helix, this sort of twisty helter-skelter shape. And these long pieces of DNA get wound up into little structures called chromosomes. So every human cell has 23 pairs of chromosomes. When we talk about the genome of a human or any other organism, we mean the entire genetic material of that organism, all of the genes put together and all of the intermediate pieces of DNA that aren't genes as well. The whole human genome has now been studied, and this is going to be really, really important for medicine in the future. The Human Genome Project was a massive undertaking by scientists in loads of different countries that ran from 1986, although it officially launched in 1990, up until 2003. They sequenced 3 billion bases from 100 anonymous volunteers to enable them to search for genes linked to different types of disease, to understand and treat inherited disorders, and to also trace human migration patterns, so looking at where genes are more similar or more different to each other, and where people have come from and how they've kind of moved across the world. In humans, sex is determined by one pair of chromosomes out of the 23 pairs that you have, and these are called the X and the Y chromosomes. If you're looking at a picture of the chromosomes, the X chromosome is a large, full-sized chromosome, whereas the Y chromosome is really, really small. Over thousands of years, it's shrunk and shrunk and shrunk until it's really quite tiny. The vast majority of people who are male will have one X chromosome and one Y chromosome, whereas people who are female will usually have two X chromosomes. That means that the sex of a baby is determined entirely by its father. The woman is always passing on an X chromosome in the egg. This means that 50% of babies are male and 50% are female if we look at the entire population of the world. In one family, it might not be exactly 50-50. And we can show how this works using a Punnett square like this. For genetics, it's quite likely that you'll get a question which asks you to either draw from scratch or fill in a genetic diagram called a Punnett square. There are two genetic disorders that you need to know detail about, so we may as well use those as examples for how to fill in this diagram. The first disorder is cystic fibrosis, and this is a disorder of the membranes where they produce lots of extra mucus which is really sticky, and so people with cystic fibrosis often find it difficult to breathe, or they have issues with their digestive system because of all the mucus getting in the way. When you fill in one of these Punnett squares, you're going to use a single letter, and it's going to be capital if it's dominant and lowercase if it's recessive. For cystic fibrosis, we tend to use F, but you're not going to lose marks if you pick a different letter. The one piece of advice I have is make sure you use something where there's a big difference between the capital letter and the little letter. So B is a really good example of a letter to use, whereas C is a bad letter to use because a big C and a little c just kind of look like the same letter, so it could be hard for your examiner to know what it is you're trying to show. So, At the top and down the left-hand side of the Punnett square, we've got boxes which represent the gametes. So here we've got two parents who are both heterozygous, and so half of their gametes are carrying the dominant allele and half of their gametes are carrying the lowercase allele. And then you're filling in the boxes so that each box contains the letter that's above it and the letter that's to the left. Once you've drawn that diagram, you want to somehow indicate on that diagram who is suffering from the disease. Because cystic fibrosis is caused by a recessive allele, it's only the people who have two recessive alleles who suffer from the disease. So if I was going to draw this here, it's in a different colour, but I might put a circle around it and say, this person is suffering from the disease. And then it's quite likely that the question will also ask you for a ratio or a probability that someone has that disease. So here I'd be saying 1 out of 4 of the offspring has the disease. Our other example of a disorder that's caused by genetics is polydactyly. And this is a disorder where people have extra fingers or extra toes. It's caused by a dominant allele. So we're going to show that with a capital letter. And what that means is that if you have that disorder, chances are you're heterozygous, and therefore 50% of your offspring are also going to have the disease. Embryo screening is a technique used by doctors with prospective parents who are worried that their baby is going to have a particular genetic disease that runs in their family. It can be used to look at a very early stage of pregnancy, often when the embryo is actually formed in a petri dish from IVF, to see whether that embryo has this particular genetic disorder that they're worried about. The point of doing it is to make sure that we're not bringing babies into the world who have a disease that are going to really, really suffer. And also, treating those disorders can often be incredibly expensive, so there's something in there about reducing costs as well. Often, when this is done, the parents will undergo IVF and they'll produce a lot of different embryos. And the leftover embryos can also be used for scientific research or even for treatment, like looking for stem cells. There are some disadvantages here. So obviously there are some big ethical issues. If you've got a lot of embryos and some of them have the disorder, those embryos are going to be rejected and then they're going to be destroyed. And some people are okay with that because really we're just talking about a very small bundle of cells, but some people believe that life begins at conception and therefore they think that that's not okay. There's the potential for people to sort of have designer babies. So people are worried that maybe parents would say, well, we want to screen our baby and see whether it's going to have blue eyes. At the present time, you're not allowed to do that. So in the UK, you can only even screen for gender if it's because there's a disease that's really closely linked to sex. So there are some diseases that are caused by the sex chromosomes, and therefore you can only have that disease if you're male, or you can only have that disease if you're female. And so you could use embryo screening and say, well, we're only going to have female babies because we know that if we have a male baby, there's a chance that he would have this disease that runs in my family. There's also an implication here that the babies that would have the genetic disease are sort of undesirable, and there's a risk that by using embryo screening, we're then sort of saying that people who are born with that disorder, they're somehow less good, and that could kind of lead to prejudice. Also, the embryos can't consent. They're just a little ball of cells. They're not even a baby yet, so there's an ethical issue there as well. Embryo screening tends to be quite an expensive process, and if you're doing the version where you're screening a baby that's actually inside the mother's uterus, there is a risk of miscarriage. And there's also a risk of false positives. So there's a chance that you could tell a woman who's pregnant with a baby that that baby is going to have a genetic disease, and she might decide to terminate that pregnancy. But there's a very, very small chance that the baby doesn't actually have the disease in the first place. This is a prime example of a question that could ask you to evaluate. So for instance, we could look at 2 different types of embryo screening and say, well, which is the better one? And in an evaluate question, you always need to be making comparisons. So for instance, saying technique A is cheaper than technique B. We want to be using those comparative words, not just saying it's cheap, but saying it's cheaper. And also don't forget that any question that asks you to evaluate, there will be a mark that is for a conclusion, and you have to write a conclusion or you don't get one. So you have to say this technique is better. You could also have an evaluate question that asks you to think about when it would be appropriate to use embryo screening. So for instance, we could talk about those 2 diseases you know about, cystic fibrosis and polydactyly. And what you might suggest is that cystic fibrosis is something that's going to potentially shorten someone's life, it's going to cause them a lot of pain and discomfort, and so it might be more appropriate to use embryo screening for a disease like that than something like polydactyly, where yes, you might have extra fingers and toes, that might make life a little bit awkward, but it's not actually going to be life-threatening, it's not actually going to cause you a lot of pain, and so it will be far less appropriate to say, well, we don't want to have a baby with extra fingers and toes, than to say, well, we don't want a baby that's going to suffer and be in pain. In order to understand evolution and adaptation, it's important you understand that we need variation in the population for this to occur. If you have an army of clones, you're not going to see any evolution. So variation means differences between individuals of the same species. If we take a room of humans and look at all the ways that they're different to each other, that's variation. Now, some variation is caused by genetics, so something like your eye colour is determined by the alleles that your parents have passed on to you. Some of it is environmental, like the presence of a scar or whether you can speak a language. But a lot of variation is actually a combination of genetics and the environment. So, for instance, how much you eat and how much you exercise obviously plays a huge role in how heavy you are, but there is also a genetic component to that. Some people have a metabolism which means they're more likely to put on weight. Genetic variation arises because of mutations, so little differences that happen in the DNA when it's being copied, or sometimes just because of mutagens, so things like UV radiation can cause your DNA to mutate. And this mutation is happening all the time. Most mutations actually have no effect on the phenotype whatsoever, but sometimes they will have an effect. And if the new phenotype is desirable, it's possible that evolution will cause it to increase in number in the population. The term evolution refers to the change in inherited characteristics over time through the process of natural selection. It may result in the formation of a new species if two populations of a species evolve so that they're so different to each other that they can no longer interbreed to produce fertile offspring. So fertile offspring are where they can then go on and have babies of their own. We now believe that all species evolved from simple life forms that first developed more than 3 billion years ago. In the exam, it's quite likely that you could be given some information and then asked to explain how a particular characteristic has evolved. Here we're going to use antibiotic resistance as an example of this. So for any type of evolution, we're going to start off with some genetic variation, because otherwise everything will just change in the same way and no evolution happens. So this genetic variation is caused by mutations. So in our population of bacteria, one of them has undergone a mutation to become more resistant to an antibiotic, which means the antibiotic is not going to be very good at killing it. So we then talk about how this means that they're going to survive more. So for the bacteria, the ones that are susceptible, when you start taking the antibiotic, the drug kills them very quickly, whereas the ones that are resistant are able to survive. Crucially, they're able to survive and reproduce. If you can't reproduce, it doesn't matter how long you survive for, you're not going to produce more offspring to pass on the allele to. So this is why some quite counterintuitive things can evolve, like a peacock's tail. It's this huge, brightly coloured tail. It makes it much more likely that it gets killed by predators, but if it can survive, it's far more likely to have babies because the female thinks it's so attractive. So therefore, it's able to evolve. So in this instance, the bacteria that have survived you taking the antibiotic, they reproduce by binary fission, and all of their daughter cells are also resistant to the antibiotic. They've passed on the allele to the offspring. As this goes on, because they now don't have any competition from the susceptible bacteria which died, they're able to massively increase in number, so as a proportion, the number of bacteria that are resistant to the antibiotic hugely increases. Selective breeding, which we sometimes call artificial selection, is the process of breeding plants or animals for particular genetic characteristics. It's basically how farmers make evolution happen really, really quickly so that they can get a new crop or a new herd of animals which have the best characteristics that are going to make them the most money. So the first point is that we need to start with a population with some variation in it. And from that population, we're going to pick the best animals, obviously a male and a female, so that they can be bred together. And they're both going to have the desired characteristic. So that could be cows producing lots of milk, or pigs producing lots of meat, or sheep producing lots of wool. So you're going to pick a really good male and a really good female and breed them together. And they're going to have lots of offspring. And then out of those offspring, you're going to pick the ones that have the desired characteristic. Because with sexual reproduction, we do have variation in the offspring. And then you take those offspring and you breed those together. And then you look at their offspring and see, well, which ones of those have the desired characteristic. And you're going to repeat this over several generations until you have a population that all have this characteristic that you're trying to get. Genetic engineering or genetic modification is an even faster and more efficient way to develop a population that have a particular characteristic. So rather than just taking two parents who have the characteristic and hoping that their offspring will inherit it when they're bred together, we can actually take the gene and directly insert it into a new individual. And this also makes it possible to transfer genes from completely different species. Plants have been genetically engineered to make bigger fruits, but also to be resistant to different diseases where that particular species isn't currently resistant. Also, it's been used for things like genetically engineering bacteria to make human insulin, and this is really important for treating diabetics who need a constant supply of insulin. In terms of the process of how this is done, special enzymes called restriction enzymes are used to cut out the required gene from the organism that already has it. This gene is then inserted into what we call a vector, like a bacterial plasmid. So this is basically another piece of DNA, and it's just going to make it easier to get the gene that we're interested in into the organism we're trying to transform. So that vector, which could be a plasmid, is put into the organism we're actually interested in, and this is done at a very, very early stage, because basically, if we do it when there's just a single cell, then when that organism grows up, every cell in it will be transformed with the new gene. Whereas if we waited until it was already an adult, then only part of the organism would be transformed. It's quite likely that you could get a question about the advantages and disadvantages or ethical implications of genetic modification. So firstly, let's talk about the fact that genetic engineering is really, really precise. We're moving one gene and we know exactly what that one gene does. It makes one protein, can't do anything else. So in many ways, there's less risk involved than traditional breeding, where we're mixing up thousands of genes in one go. However, there are still some concerns. So we're mainly thinking about crop plants, things that we're going to eat. So sometimes these are transformed with herbicide resistance, so that you can spray a whole field with a herbicide that's going to kill weeds and it won't kill your crop. And this obviously means it's far less time consuming than if you're going around spraying the weeds individually and trying not to spray the food. But there are concerns that the crop plant could then breed with weeds, and then the weeds could become resistant to the herbicide, and then that would obviously be really, really bad. There are also other crop plants that have been transformed with some kind of poison like cryoprotein to basically stop things like caterpillars from eating them. But then there are concerns about whether this could impact pollinators and things. And for both of these situations, there's relatively limited research done into whether there could be any kind of adverse impact for humans eating these crops. So there are concerns there. Then also people have started talking about genetically modifying humans. So there are various kinds of gene therapy where you try to overcome some kind of genetic disease by essentially genetically modifying a human. And there we really start to get into the ethical grey areas of, are we playing God? Is it okay to do that? Is it better to relieve someone's suffering, or should we just not be interfering? So you need to be prepared to discuss those things. And remember, if you get a question that says evaluate, it's really, really important that you write a conclusion. When we say that a species has gone extinct, like the western black rhino in 2011, we mean that every single individual of that particular species has died out. There are no more and they're not coming back. This could happen because of a new disease, a new predator, and here we do include humans and hunting, a new competitor that's better at getting the food, a natural disaster like volcanoes, temperature change, which is particularly applicable if you're thinking about plants and their moving ranges, or insects that are not able to move their range as quickly as the plants that they feed on or pollinate, and also something called the cyclical nature of speciation, which basically means that over very, very long periods of time, one species ends up turning into a completely new species. And so the first species has effectively gone extinct because there are none of that old type of organism left. When we're trying to learn about extinct species, the fossil record is really, really important. And it's really useful for studying evolution because we know that the deeper down in the rock we find a fossil, the older that rock probably is, and therefore the longer ago that species was alive. So when we say fossils, we mean the remains of an organism that have been preserved, usually in rock, but sometimes in ice or in amber, and usually over millions of years. Most of the fossils that we have are the rocky type, and they form when an organism dies and then layers of sediment, so sand and mud, have formed on top of the remains. And that's usually going to happen under the sea. Because oxygen isn't able to get in, bacteria aren't able to decay things. So over a very long time, the hard parts, like the bones or the teeth, can become mineralised. So minerals take the places of the bones, and basically we end up with a rock that has the shape of a bone or something in it. And this kind of explains why we don't really have that many fossils of things like jellyfish or worms, because they didn't have hard parts to fossilise. It's also possible to fossilise the remains of something. So rather than just the animal, it could be its droppings, or it could be a footprint, or it could be a burrow. Classification is the science of putting things into groups based on their shared characteristics, and you need to know about two classification systems. The old school taxonomic hierarchy system, which we used for a very long time, and then also the three-domain system, which was proposed by Woese and his colleagues in 1990. So in this new three-domain system, we have the eukaryotes, which includes us humans, but also plants and yeast and anything else that has a nucleus. Then you've got the true bacteria, and finally you've got the archaea, which are actually as different from bacteria as they are from us. But according to your exam board, they're primitive bacteria, usually found in extreme environments. So there are a lot of extremophile archaea. Now, in the old system, we had 5 kingdoms. So this was your animals, plants, fungi, prokaryotes, and a group called Protoctista, which sort of has algae and slime molds, and also Plasmodium, which causes malaria in it. And then each one of those kingdoms is split into a number of phyla. So for instance, our phylum as humans is called the chordates, and that includes everything that has a spine and a backbone, as well as a few other things that don't. Then we split that down further, so we have our class, and our class as humans is mammals, so that can be defined as organisms that produce milk, and we have body hair, and we give birth to live young. Then that's split further into an order, so ours is primates, that includes all of the sort of monkeys and chimpanzees and things like that, and then down into a family, and ours is hominids, so things that are kind of human-shaped, basically. And finally, we have a genus and a species, and the genus and species are used to make up what's called the binomial name. So sometimes people talk about this as a Latin name, but actually loads and loads of these names are not Latin, so you shouldn't call it that. It's called the binomial name or the binomial system. So every species has a binomial system which has 2 parts. The first part is the genus, and the second part is the species. So for instance, you'll have heard humans called Homo sapiens. Homo is our genus, and there are other species that are of the Homo genus, and then sapiens is our species name. An ecosystem is the interaction of a community of living organisms, or the biotic factors, with the non-living or abiotic parts of their environment. To survive and reproduce, organisms require a supply of materials from their surroundings and from the other living organisms that live there. Animals and plants may compete either within their species or intraspecifically, or between different species interspecifically, for both the biotic and abiotic factors. So where an organism is able to live and survive and thrive is going to be based on a combination of biotic factors and abiotic factors. The biotic factors could be things like the availability of food, so either prey or plants and fruit if it's herbivore, predators which are going to try and eat you, competitors for that food and also for things like shelter and mates, whether there are diseases, and also how easy it is for you to find a mate. It's no good you finding the perfect environment where you can survive for ages if you can't actually have any babies there. And then the abiotic factors for plants, light intensity is obviously going to be a big one, so they can photosynthesize, the temperature, the moisture levels, the mineral ions, the soil pH, and also carbon dioxide levels. And then if you're an aquatic organism like a fish or a newt, then oxygen levels in the water are also going to be really important. When we talk about interdependence, we're talking about how species depend on one another for survival. So for instance, maybe insect A drinks the nectar of plant C as it's pollinating its flowers, and mammal B eats its fruit. And therefore plant C is dependent on insect A to pollinate it, and also mammal B to disperse its seeds, which are inside the fruit, after it's eaten them. This is called interdependence. And in a stable community, all of the species and the environmental factors are in balance so that the population sizes remain fairly constant from year to year. Over time, species are going to become adapted to the environments they're living in. And here we're talking in evolutionary terms. We're not saying, oh, this one individual bear grew a thicker coat. We're saying over thousands of years, bears that have thicker coats have survived better and had more babies, and therefore, in general, the bear's coat has become thicker. So when we say adaptation, we mean a structural or behavioural modification that has evolved, often over thousands of years, to give an organism a survival advantage in its habitat. This comes up quite regularly as a question where you're asked to sort of give some examples of adaptations and explain them. So we could have things like if you're living in the desert and you're a plant, you're going to have small leaves, maybe even spines, fewer stomata, big roots to get all the water. If you're an animal, you might have big ears to allow you to sort of disperse some of that heat. And behaviourally, you might only come out at night and you might be nocturnal because it's colder during the nighttime. If you're living in the Arctic, you might have thick fur and blubber for insulation, you might be camouflaged, and you might have big paws to spread your weight over the ice. In terms of avoiding predation, we've got the use of spines to stop animals eating you if you're a plant, poisons and also stings, using warning colours, and that could also include mimicry. So you've got things like a hoverfly, which pretends to be a wasp. It's got that yellow and black colouring, and so other animals don't eat it because they don't want to get stung, but actually it doesn't have a sting of its own. And also camouflage, so things like a stick insect, making itself look like a leaf or a stick so that it doesn't get eaten because the predator just doesn't see it. Within an ecosystem, we're going to have various feeding relationships, and we can show these using food chains or food webs. So in the food chain or food web, the arrow represents the transfer of energy. So it always goes from the thing that's being eaten to the thing that's eating it and taking the energy. Every food chain or food web starts with a producer, which is something that can make its own food. And for the purposes of GCSE, this is always going to be something that does photosynthesis, so a plant or an algae. So all of the energy in all of the food chains starts with the sun, and then plants use photosynthesis to lock that up in glucose. Anything that isn't a producer and needs to eat its food is called a consumer, and we give them different names based on which trophic level they're at. So herbivores, which eat producers, are called primary consumers because they're at the first trophic level, they're the first thing that does the eating. So here in this example, our zooplankton and our krill would be primary consumers because they are consuming the algae and the phytoplankton. Then the secondary consumer is something that eats a primary consumer, so it could be our big fish here. And a tertiary consumer is something that eats a secondary consumer. Now you can use this to start talking about how would the ecosystem be affected if one thing was taken away, or if one of these organisms went extinct. So for instance, if our krill suddenly went extinct, the number of phytoplankton might increase because there's one fewer thing eating them. And the number of fish might decrease because the fish don't have one of their main prey. It's also important we're thinking about the abiotic processes, including the carbon cycle. So the carbon cycle is all to do with everything that is changing the amount of carbon dioxide that's in the atmosphere. So we've got some processes that release carbon. All living things do respiration, so that's your plants and your animals and your fungi. You've got your decomposers doing decomposition, where they break down animals and plants after they've died, and also the combustion of fossil fuels. Then we've got processes that take in carbon. So photosynthesis is obviously the big one, taking in all that carbon dioxide. You've also got carbon dioxide dissolving in the oceans and being used by aquatic organisms to make shells and make carbonates. And also, in a very long-term scale, we could be thinking about the production of fossil fuels or peat, which are also going to lock up carbon. And then we've also got carbon just being transferred around other members of the community. So for instance, whenever an animal eats a plant, it takes the carbon that was in the plant's body and puts it into its own body instead. Biodiversity is all about having a lot of different species, either on Earth or in a particular ecosystem. And the reason we care is that the higher biodiversity is, the harder it is for that ecosystem to crash. Basically, if you have a system where you maybe only have 2 or 3 species and they're completely dependent on each other, if something bad happens to one of them, then everything else comes crashing down too. Whereas if you've got lots and lots of different species and maybe everything has 5 or 6 different sources of food, then even if something bad happens to one population, everything else is going to be able to stabilise and recover. There are lots of different things that we as humans can do to protect and encourage biodiversity. So this includes things like breeding programs for endangered species, protecting and regenerating rare habitats, trying to reintroduce field margins and hedgerows around farms, because just having one species in that place is just not great for biodiversity, reducing deforestation, recycling things, and also conserving peat bogs. In terms of other things that humans are doing that are really bad for the environment, there's obviously a lot of pollution being caused, which kills animals and plants and reduces biodiversity in terms of water, air, and on land. By us building, quarrying, farming, and dumping waste, we're reducing the amount of land and habitat available to animals and plants. And also we've carried out large-scale deforestation in tropical areas, such as the Amazon rainforest, both to provide land for farming, for cattle and for rice, and also to grow crops for biofuels. The greenhouse effect is a natural phenomenon which stops Earth from being so cold that nothing can live here. It's caused by gases like carbon dioxide, methane and water vapour, which form a blanket around the Earth's atmosphere. Shortwave radiation from the sun, like visible light, is able to easily penetrate this blanket and get to Earth, but when it gets here, it's absorbed and re-emitted as longer wavelength radiation, like infrared radiation, which can't so easily get through the blanket of greenhouse gases. Therefore, this just stays in our lower atmosphere and it warms up the atmosphere, and therefore Earth is getting warmer. And the problem we have is that humans are producing more and more of carbon dioxide and methane, both from burning fossil fuels and from farming cattle and farming rice, and so the greenhouse effect is getting stronger and stronger. This is leading to rising temperatures, which is going to lead to things like extreme weather events like hurricanes, melting ice caps, flooding, habitat destruction, and eventually extinction of other organisms. The second required practical in Biology Paper 2 involves us measuring the population size of a common species, and often this will be in the context of us looking at how it's different based on an abiotic factor. For instance, is there more clover where there's lots of light or where there's less light? So you're going to need to use this piece of equipment here, which is called a quadrat, and the idea here is that we lay it down and we count to see how many of the squares contain the species we're interested in, and we can use this to work out percentage cover. So we can either do this by random sampling or we can do this by transect sampling. So in random sampling, you might have done this where you sort of shut your eyes and you throw the quadrat over your shoulder, or you might have done it with a random number generator. So the way this works is that you use two big tape measures to basically lay out a big grid on the area you're sampling, and then you use something like Excel or a different random number generator to produce you a list of numbered squares you're going to go to. So you go, okay, so we're going to go to B1 and sample there, and then we're going to go to C5 and sample there, and you do it that way. Obviously, if we're trying to compare this to an abiotic factor, we need to be taking a light reading or a pH reading each time that we sample. And it may be that we're going to do this in multiple different areas. If we're doing the transect sampling, then you're going to use a tape measure to measure out a really, really long straight line, and you're going to go along and put your quadrat down at regular intervals and see what the difference is there. So that could be something like starting in a woodland and moving out into the sort of the open air as there's more light, and seeing how that affects it. I really hope that was helpful for you. Good luck for your exams, and don't forget to subscribe.